Let's summarize what we've talked about with regards to DNA replication. We know that DNA will be replicated or copied anytime that a cell is preparing to divide. And in prokaryotic microbes, in bacteria and archaea, the DNA molecule is a circular double helix. So, in order to begin replication, that double helix has to be opened. The two strands have to be separated from each other from the point of origin, and an enzyme will do that. Then we need to prime each of the old strands of DNA so that new DNA can be built. An RNA primer, a short piece of RNA, will be placed on each old strand of DNA. Another enzyme is going to do that work in order to get the replication process started. At the end of that primer, you're going to have a 3' hydroxy functional group jutting off. And the DNA polymerase enzyme will see that 3' hydroxy group as the place to begin to add on the next nucleotide. DNA polymerase will build new DNA on the two old DNA strands. But remember, it can only build DNA in one direction. It can only add new nucleotides to the 3' end of the last nucleotide that was added. We're going to give this a name. We're going to say the direction of synthesis, of replication of DNA, is 5' to 3'. DNA polymerase builds new DNA in one direction, 5' to 3'. And the way to read this is to say to yourself that this means that we can only add new nucleotides to the 3' end of the existing new strand. When the DNA polymerase builds new DNA, part of each new strand is going to be made continuously, and part of each new strand is going to be made in fragments called Okazaki fragments. And the reason for that is because of the unique orientation of DNA and the fact that the old strand and the new strand have to be anti-parallel to each other. The parts of each new strand that are made continuously are made through leading strand synthesis, and the parts of each new strand that are made discontinuously in fragments are made through lagging strand synthesis. Once the entire DNA molecule has been replicated, those RNA primers will get removed and replaced with DNA by another enzyme. All of those fragments of DNA, all of those Okazaki fragments, have to have their own primer. So all of those primers have to be removed and replaced with DNA so that we will have one long continuous strand of DNA, not interspersed with RNA primers. When all of the primers are removed and replaced with DNA, all of the little breaks in the strand will get sealed up. Of course, that's done by another enzyme. And we'll end up with two molecules of DNA, two circular chromosomes. And each one will have one old strand in it and one new strand in it. Now, the DNA polymerase enzyme is really remarkable if you think about it, because it can copy an entire chromosome in really quite a short amount of time. Remember, we said that there are species of E. coli, for example, that can go through binary fission with a generation time of about 20 minutes. Think of all the things that have to happen during binary fission. It's not just copying the DNA molecule. Everything inside the parent cell has to be copied, including the DNA molecule. And that DNA molecule is being replicated in something less than 20 minutes. That's pretty remarkable for this enzyme. It works very quickly, and it is very accurate, but it is not perfect. There are no biological systems that work 100% perfect all the time. So every once in a while, the DNA polymerase enzyme is going to put the wrong base in that new DNA. In fact, it happens about one time out of every 100 to 1,000 bases that are placed. So the enzyme should be placing a T in the new strand because there's an A in the old strand. But instead of placing a T, it makes a mistake, and it puts a G there instead. About once out of every 100 to 1,000 bases, it makes a mistake. Now, the good news is that the cell will proofread any new DNA that gets made, and in fact catch most of the errors that enzyme makes. That's good news. The cell proofreads that brand new DNA, and if it finds a mistake, it will fix the mistake. The bad news is that all biological systems are imperfect, so even the proofreading system makes mistakes. Sometimes these mismatched bases remain there, and you end up with a mutation. What exactly is a mutation? By definition, a mutation is a permanent change in the order of the bases in DNA. A permanent change in the sequence of the A's and the T's and the G's and the C's. Now, you've all heard of mutations. We've talked about mutations in laboratory. We talked about how ultraviolet light can damage DNA. And when the DNA polymerase enzyme tries to copy damaged DNA, it often places the wrong bases. That leads to mutations, to an alteration in the sequence of those bases from what it is supposed to be. You probably know that many mutations that happen to DNA come from exposure to dangerous molecules. There are lots of chemicals, for example, that are capable of causing mutations in our DNA. We refer to those as mutagens or carcinogens because they can often produce mutations that then lead to cancers. You've probably also heard that exposure to ionizing radiation, X-rays and gamma rays, can lead to DNA mutations and also sometimes to cancer. We talked about, again, how ultraviolet radiation can ultimately lead to mutations in DNA. But now we're going to add that third thing in. We're going to add in that sometimes it's just errors. It's just errors during the replication process that cause mutations. Now remember, in the genetic code, the code that all living cells use to build protein, in the genetic code, groups of 3 bases, and we call those codons, are used to code for one amino acid. When a mutation occurs, we are changing the sequence or the order of the bases in the DNA. That means we're changing the order of the bases in at least one codon. If you change the order of the bases in a codon, there is at least a potential to change the amino acid that codon will call for. If the amino acid that should be there is replaced with a different amino acid because of the mutation, now we have altered the order or the sequence of the amino acids. Now we have changed the protein. And if you change the protein, you change the trait. So mutations in DNA can lead to changes in proteins, and changes in proteins change traits. Sometimes mutations are actually a good thing. Mutations are the basis of evolution. Without mutations, organisms would never develop new traits. And some new traits are really helpful to an organism. Some new traits are what we call adaptive, and they help populations evolve over time. But some mutations are not adaptive. Some mutations are harmful to the organism. As we've said, some mutations lead to cancers, disease, and even death. So let's talk a little bit about mutations in terms of the different types that we see. There are really 2 categories of mutations, what we refer to as point mutations and what are called chromosome level mutations. Now, if you hear a mutation described as a point mutation, it means that a single base has been altered somewhere in the DNA. If you hear a mutation described as a chromosome level mutation, it means that an entire region of the DNA has been changed. An entire section of a chromosome has broken off and sometimes rearranged. Now, we're not going to talk about chromosome level mutations in depth in this course because you just don't see them very often in bacteria and archaea, in the prokaryotic organisms, because they have a circular chromosome. But in eukaryotes, the kinds of organisms that have multiple linear chromosomes, chromosomal level mutations occur all the time. And as you might imagine, they can be devastating to the organism. Because again, we're not talking about one base being out of order. We're talking about entire regions of DNA, hundreds and thousands of bases being out of order. But again, we're going to focus on point mutations because those are the ones we see the most often in prokaryotic cells. Now, there are 2 types of point mutations. There are what are called substitution mutations, and there are what are called frameshift mutations. Now, let's talk about what these terms mean. When you hear about a substitution point mutation, that is describing one base again being substituted with another. So maybe we should see an A in that location in the DNA, and instead we see a C. The A has been substituted for a C. Now that can lead to a change in the amino acid, but again, only if that substitution happened in a place where that matters. Let me explain what I mean by that. Recall when we looked at the genetic code, the table, we said that amino acids are often coded for by more than one codon. And as long as the first two spots, the first two positions in the codon are correct, you won't see an amino acid change. You'll only see a change if we have a problem in those first two positions. In other words, it's the third position, the third base in a codon, that has all kinds of room for error. If I should see an A in the third position, and instead I get a C, so I went from a codon of UCA to a codon of UCC. I have a mutation there. I've changed the order of the bases, but I'm not going to see a change in the protein that gets made because I'm not going to change the amino acid. Serine is coded for by both of those codons. But what if I had a mutation in the first position? What if it went from UCA to CCA? And that's a different story. If you look through the table for CCA, you can find it right here. When we get to that codon during translation, the cell is not going to place a serine there like it should. It's going to place this amino acid, a proline, instead. So when I say that mutations can alter the amino acids, it's because it depends on where the mutations are. It's typically the first and the second position in a codon that is critical. If a mutation occurs in the first or the second position, it often leads to a change in the amino acid. If it happens in the third position, it's usually not a problem. So, a substitution mutation simply substitutes one base for another. And depending on where that base is changed, we may or may not see a change in which amino acid is being coded for. If we change the amino acid order, we're going to change the protein. And if you change the protein, you're going to change the trait. The second category of point mutations are referred to as frameshift mutations. These are a little different. They still involve a single base, but rather than substituting one base for another, in a frameshift mutation, an extra base might be added in, or an existing base might be completely removed from the DNA. As you can imagine, frameshift mutations can be devastating because when you take a base away or you add a completely extra base, from that spot onwards, you have altered every single codon. You have altered the frame. In other words, you've shifted every single codon by one base. That's referred to as the reading frame being shifted. So all of the codons past the point of a frameshift mutation will be altered. And that means, at least potentially, all of the amino acids past that point will be different. So if that happened, you would get a massive change in the protein. You would get a massive change in the trait. So there are point mutations and there are chromosome level mutations. And in the category of point mutations, there are substitution point mutations and there are frameshift point mutations. Within the category of substitution mutations, we can describe three types. We can describe, again, just that basic substitution, where one base is switched for another. That can lead to a change in the amino acid for a codon, which can then change the protein and change the trait. Now, there is a second kind of substitution mutation referred to as a silent mutation. In a silent substitution point mutation, we are still switching one base for another because it's a substitution type, but we are absolutely not changing the codon. We're changing the codon, but we are not changing the protein because we're not changing the amino acid. So silent mutations are those mutations, again, that tend to hit the third position in the codon. It's going to change the codon because you've changed the order of the bases. But that codon will still code for the same amino acid. So the mutation is thought of as being silent because it has no effect for the cell. It puts the same amino acid in that position, you get the same protein, you get the same trait. It's referred to as being silent because it has no impact. Finally, for substitution mutations, we have nonsense mutations. Again, it's a substitution, so we're switching one base for another. It just so happens that that switch turns an ordinary codon into a stop codon. Remember, there are 3 stop codons. And normally, a stop codon occurs at the very end of that protein coding sequence. So if I mutate a base and I create a stop codon where there shouldn't be one, that's going to cause the whole process to stop too early. What that often does is it creates a prematurely shortened length of amino acids. So you're going to get a very abnormal protein from that